practice, SARS-CoV-2 spreads through droplets that are released when an infected person coughs or sneezes. These droplets can travel for 2 meters, so they can easily be transmitted to people breathing in those droplets. The droplets can also land on objects and survive between several hours or even days. A person can also be infected by touching his or her face after touching those objects. To protect yourself and slow down the transmission, clean your hands frequently with hand sanitizer or soap and water for at least 20 seconds in order to break apart the virus. Cover your sneeze into your elbow or use masks to prevent the transmission. So whenever you're going outside, use a 3-layered fabric mask correctly to ensure its effectiveness. But we have to keep this in mind. Masks are not replacement for physical distancing, hand hygiene, or other public health measures. So do yourself a favor by staying at home. Why? Because the incubation period for the virus itself is from 2 to 14 days. This means you could be infected and show no symptoms between this period. If you're practicing physical distancing, you would break the chain of infecting others and slow down the transmission. Keep yourself healthy by eating fresh food, doing some exercise, sleep properly, and continue to follow respiratory hygiene practices. If you experience any symptoms, isolate yourself from others for 2 weeks. If you continue to have symptoms, seek medical attention or call COVID-19 hotline provided by the government. This isn't just about medical workers or police anymore. We have to unite and fight this hand in hand to overcome this pandemic situation. Stay at home and be hero for others.
Hundreds of years ago, the world had been hit by several pandemics. Some of the worst pandemics recorded in history are the Justinian's plague, Black Death, and Spanish flu. Now, a new pandemic has been discovered in Wuhan, China. More than 6 million cases and 300,000 deaths have been reported from all over the world. It's called COVID-19. COVID-19 has been reported for the causative agent of pneumonia. Furthermore, this virus is identified as a member of the coronaviruses group called SARS-CoV-2. What is coronaviruses? Coronaviruses are a family of RNA viruses. They are called coronaviruses because the appearance of its spike protein resembles a crown. There are several of them that have been made an outbreak, such as MERS-CoV, which is transmitted from the dromedary camels to humans. What about SARS-CoV-2? SARS-CoV-2 is transmitted from human to human by the respiratory droplets when we are talking, coughing, and sneezing. The transmission from human to animal considered to be low as well as animal to human, and the research is still going on. Although genetically closely related viruses have been isolated from rhinolopus bats, the exact route of transmission hasn't been established yet. Now, the question is, how about COVID-19 infections in animals? Some animals have high susceptibility such as cats, ferrets, and tigers, low susceptibility like dogs, and not susceptible like pig and some poultry animals. According to OIE, cases of animal infections have been reported by several countries such as mink farms in Netherlands, cat, lion, and tiger in USA, dog in China, and cat in some countries. The information about the transmission route is still limited. However, as for the other respiratory viruses, it appears that the virus is transmitted from direct contact like droplets. How can we make contact with animals in safe ways? First, don't let animals interact with other people or other animals outside your home. If a person inside your home is sick, isolate he or she from other people including animals. Always wash your hands with soap and water for at least 20 seconds before and after contact with animals. What should I do if my pet or animals are showing COVID-19 symptoms? Do not take your pet or animals to the veterinary clinic by yourself. Please kindly call your vet. Your vet can then evaluate your pet or animals and determine the next steps for the treatment and care by phone or seeing them by themselves.
Okay, it looks like all the speakers and all the attendants already been YouTube. Okay, good afternoon everyone and in Indonesia and good morning and good evening for everyone around the world. Before we start, let me introduce myself. My name is Krisnalini Andrian, people call me Ali, and I am a staff of One Health Collaborating Center Universitas Gadjah Mada. I hope everyone right now at a good condition. Welcome to our webinar that has been held today. A very interesting topics and an expert speaker at this expertise. We held webinar because we knew that many diseases not yet to be known, especially that came from wildlife. We know too that the identification procedure need to be quick, specific as the technology and the science is progressing. We need to keep update our knowledge about that. And today, we hope that this webinar will give everyone a new understanding or maybe a more deeper understanding about novel technology, wildlife identification. Okay, and let me share my screen. Today webinar will be titled Novel Technology for Wildlife Pathogen Identification. And this webinar schedule will be a welcome speech from Professor Dr. Wayan Tunas Aktama, DBM, the One Health Collaborating Center coordinator, and then will be continued by the first topic delivered by Dr. Ronald Runtuwe from National Institute of Infectious Disease, Japan. His topic was New Paradigm of Molecular Identification of Zoonotic Pathogen. Then they will be continued by the second topic delivered by Dr. Johan Michaux, Ph.D. He's from University of Leige, Belgium, with his topic Microbiome Analysis in Wildlife. There will be a question and answer session too, and then there will be a closing. In the closing, I hope that Mr. Ryan and Dr. Luki and Dr. Johan will give a closing statement. Okay, for the first session, I invite Professor Ryan to give a welcome speak and open this webinar. For Prof. Ryan, the time is yours. I'm sorry, Prof. Ryan, you are still mute.
Thank you so much for the time. Good morning, good afternoon, everybody, especially for our distinguished guest, Johan in Belgium. Now it's still morning over there. And Luki in Japan is already afternoon, late afternoon. So I do hope that both of you are healthy and want to share with us your experiences in molecular new paradigm in molecular detection for Luki, and microbiome analysis in wildlife for Johan. I think the first thing for the One Health approach is identify the pathogen, and then second is mitigation of the risk, and then how do we can do a early response, early detection, and then how to minimize the human risk reduction. This is the point that we want to share for today for all of us. Thank you for your attendance for the our last seminar, last web seminar series this December, and maybe see you again in next year in January 2021. Okay, thank you. Have a nice discussion. I hope that all of us can get a new view of the molecular detection in wildlife and also in the microbiome identification. Thank you so much. Thank you. I give back to the Noli. Thank you so much.
Okay, thank you, Parvayan, for our welcoming from Parvayan. And then in this webinar, there will be rules too. The first one is participants may ask the speakers during the webinar for the question link. The question link will be on the chat room too, and the other issues might be asked in the chat room too. Your attendance will only be confirmed by filling the online form shared at the end of this webinar. And then the e-certificate will be charged according to the attendance form and emailed about 1 week on the webinar. And then the materials will be shared with the okay. In this webinar, we will be led by a moderator, and let me introduce her. Our moderator will be Ms. Putu Devi Cakalandiswara, DVM MSc. She is staff and research assistant in One Health Collaborating Center Universitas Mada, working with genomic study, microbiology, zoonosis, and antimicrobial resistance in agriculture environment. Her education was DVM program in Faculty of Veterinary Medicine, Universitas Mada, Indonesia, and then Master of Science, Veterinary Science and Technology, Faculty of Veterinary Science, Ronald University, Thailand. Right now, she is at doctoral program in biotechnology, graduate school in Gadjah, Mada, Indonesia. Her organization was Infectious Diseases Research Unit, FIDSRU, Department of Microbiology, Faculty of Veterinary Science, Ronald University, Thailand, from 2017 to 2019, and One Health Collaborating Center Gadjah, Mada, from 2016 until now. For Ms. Putu, the time is yours.
Thank you, Noli, for your warm introductions. Hello, everyone. Thank you very much for joining. Hello, Dr. Luki. Thank you, Dr. Johan. Thank you very much for accepting our invitations. Today, we will learn about the novel technology for identification of pathogen in wildlife. And maybe somebody asks, why is it important and why have to be wildlife? As we know from the last webinar, that the presence of emerging infectious diseases are dominated by the zoonotic disease. It's around 60%. And from this number, 70% are originated from wildlife. As we know that emerging infectious disease is those diseases that never happened before in a very long time ago, or it's just appeared and becoming outbreak. And because this is the emerging infectious disease, most of them are caused by the pathogens that never identified before, or it's a novel pathogen. As Prof. Wayan already mentioned, that we need to identify those pathogens as early as we can, so we can make the mitigations. And in the other hand, somebody of us already heard about the novel technology such as the next generation sequencing, and then the metagenomics, the microbiome, this is the experts of today's our speakers. And that's why we come here and we invite both of our speakers, and we will learn about it together and discuss it as well. So let's move for the first speakers. Dr. Luki, are you ready? I will introduce you first before you start your presentation, so that our participants can can know where you come from. So Dr. Ronald Runtuwe, he is a senior researcher in AIDS Researcher Center, the Research Center, National Institute of Infectious Diseases, Japan. He is a visiting researcher in Graduate School of Frontier Science and in the University of Tokyo. He finished his medical doctor in Samratulangi University, Manado, Indonesia. He is a research student when he was in Oita University, Japan, and he finished his PhD in medical science in Oita University, Japan. And he got several grants or achievements, such as Hirose International Scholarship Foundation from 2019 up to 2020, grants in aid for scientific research, Kakeni 2018 until 2019, GSPS, CSPS Research Fellowship for Young Scientists, DC2 from 2014 to 2016, and Tetsuya Otaki Award from Japanese Society of Medical Entomology and Zoology at 2014. So I will invite Dr. Luki, and the screen is yours. Thank you very much.
Thank you so much for the kind introduction. Can everybody hear me?
Yeah, we can hear you perfectly.
Clear.
Okay. Prof. Wayan, how are you?
I'm fine. Thank you, Luki. Thank you for joining the seminar.
Yeah, thank you for inviting me to give to share some of my knowledge, all of you. So let me share my screen first.
You forget to mention the GSPS that we get together.
Sorry?
GSPS. This is on the seminar program from GSPS that we have.
Oh, okay. Okay. Thank you also for the GSPS for supporting this seminar.
Yeah.
I wish the Komodo trip able to be conducted this year. Apparently, because of the coronavirus, it's impossible.
Yeah.
So we can only meet each other in online. But it's been a great pleasure. Okay, so let me share my screen first. Okay, can everybody see this?
Yeah. Yeah.
So my talk is about new paradigm for sequencing technology in zoonotic research. So I changed a little bit. So I was tasked to give like new paradigm for pathogen identification, but I think I will, because I've been working with sequencing technology for the 5 years. So I will talk a little bit about sequencing technology and how it applies in zoonotic research. So not only in pathogen identification. So so the topic would be like a little bit wider for today. So as have been introduced before, my name is Luki Runtuwe. I'm originally from Manado, Indonesia, but now I work in the AIDS Research Center in National Institute of Infectious Disease in Japan as a senior researcher. I'm also a visiting researcher in the Tokyo University. Okay, so for the talk today, these are the outlines of the talk that we will be discussing today. Is first about how sequencing is always an evolving tool for molecular research, and for some who doesn't really know what sequencing is, I'm gonna talk a little bit about it. And then we will go deeper into sequencing technology, which is the high-throughput sequencing that encompass Illumina sequencing and other stuff about it. And also for a more proper application in developing country and also in field sequencing, I will talk, the later part of my presentation will focus on long-read sequencing, such as the nanopore sequencing. And also, I also will talk a bit about our project with nanopore sequencing that we've been performed. Okay, so this is a change of paradigm in molecular research. So ever since the discovery of DNA, many researchers focus on how to manipulate the DNA and genes. But before 1980s, before the discovery of PCR, working with DNA was actually very difficult. So although the sequencing technology was actually found before the PCR, which is sequencing technology found in the 1970s. However, to do the sequencing at that time is like very difficult, and also so for example, if you want to amplify a gene of interest, we have to like cut the gene of interest using precision enzyme, and also have to clone it using bacteria system, which was considered practical way to amplify gene of interest before 1980s. And then on 1983, PCR was discovered, and then molecular research just seems to like explode exponentially up to this time. Now we can actually very easy to apply to amplify gene of interest. We can actually very easy to read this sequence. We can edit the gene of interest. We can make proteins from the gene of interest out of the body, and then we can actually administer the protein of interest as a vaccine or drugs, which is actually can be done now in COVID vaccine, such as the vaccine for Moderna. And so for those purpose that actually therapy can actually be personalized. So we can actually read somebody else genome. We can actually make drugs based on the genome of the person. And to be able to have such a personalized therapy like that, we need sequencing technology, because that is the only way we can read the DNA of the genome of somebody or a pathogen. So sequencing technology now actually has the power to lead to the precision medicine. And okay, so what is sequencing? So in the simplest term, sequencing is actually a method to read the sequence of nucleotides in our DNA and our RNA. So for example, this everybody probably knows what is this figure. This is the figure of DNA. Adenine pairs with thymine, cytosine pairs with guanine in a double helix formation. So sequencing technology will try to actually read this sequence of this ACTG. And the sequencing technology was developed on 1977 by a researcher called Frederick Sanger. We hear his name a lot now as a Sanger sequencing, because he was the one that discovered the practical way to sequence DNA. So the original sequence is actually called chain termination method. And to be able to do that, we actually have to do like the original sequencing method is actually pretty cumbersome also. So you need about 4 reactions for each strand of DNA that you want to sequence. And after you run the reaction, you have to load it into a gel. For example, this is the example on how you read a DNA from the original sequencing technique. So if you what each lane of this one is contains the chain terminator of ACTG. And if you want to read this one, you can read this. This line is A, is correspond to adenosine. This one correspond to cytosine. This one correspond to thymine, and this one correspond to guanine. And if you want to read this DNA, it will be read it from by counting on the band. So this DNA would be read it as GGG, CGGT, CGT, etc. So this is like the original sequencing technique that was performed on the 1970s. And the technique was actually used to sequence like smaller genomes such as virus, yeast, worms. It was very easy to do because those only have like very small genome size. But on the 1990, this technique was actually used to finally to read the sequence of a human genome. And on the 1990, a consortium called the Human Genome Project was constructed with only the sole purpose of sequence the first human genome, which human genome contains about 3.2 gigabase. But at that time, they actually need about 13 years to finish the whole human genome, because it's a very complicated process at that time. next generation sequencer was not developed at that time, so they have to do everything manually. They have to do it like with two parts of a process. The half part was about establishing the physical map of human genome, and the later part was actually to sequence the genome by itself. So at total, this is a graphic that shows the timeline of the human genome from the 1990, and it takes about 10 years only to finish the draft of a human genome, and another 3 years to finally finish the human genome. So you can see that at time it's very difficult to do the sequencing DNA. However, on the 1990s, also the original Sanger sequencing was actually automated. So we actually, probably most of you have seen this machine. This is called the ABI DNA analyzer. This is actually based on the original sequencing technique, but this has been automated. So after the discovery of capillary electrophoresis, and this machine transfer this electrophoresis into capillary and actually use a laser to excitate the DNA. So now, even though they have the same principle, but it's been automated by using the laser system in this ABI DNA analyzer. And this machine is, even though this is like a very old technique, but this machine is still used now, because this is a very accurate. The result from a DNA analyzer is very accurate, although it's like, and also very cheap, but it has only like very low throughput. So we can only sequence up to like 3 kilobase of DNA strand. So then came the revolution of DNA sequencing on the year 2000. So high throughput sequencing came out. So at the year 2000, there were about 3 major companies using the sequencing technique, which is the one is the Illumina genome analyzer. At the time, it was called Solexa genome analyzer, actually, before it was bought by Illumina. And also there was also a machine from ABI, which is called ABI Solid, and also Roche also produced a sequencer machine, which is called the Roche 454 Pyro sequencer. So at the year 2000, these 3 companies produced the high throughput sequencing. And what's different with the high throughput sequencing is actually it changed how DNA sequencing works. So, for example, if you can see here on year 2001, it takes about $100,000 to only sequence one genome. However, by the time this machine came out, the cost and also the data produced is actually exploded exponentially. So on year 2008, the human genome was resequenced using Illumina platform. And by year 2004, about 228,000 human genome was already sequenced. And to sequence one human genome now, it only costs about $100,000. So you can see that because the revolution of this machine, now DNA sequencing has actually become very easy to do. And let me talk a little bit about application of high throughput sequencing. So there are a lot of applications that you can do with Illumina platform, such as sample genomics. This is the simplest one. You can read a DNA of a genome of a pathogen or a host. There are also transcriptomics, which is you want to learn about gene expression. So in this case, you sequence the RNA. And there are also applications in epigenetics, metagenomics, microbiomics, single cell transcriptomics, and long read sequencing. So these are the major applications of high throughput sequencing that has been the Illumina platform can be performed. So let me start a little bit about transcriptomics. So transcriptomics research is actually mainly focused on the expression of genes. So the central dogma of biology is from DNA. It will be transcribed into RNA, and RNA will be transcribed into protein, and so that's how the gene will be expressed. So depending on condition of depending on different condition or different experiments, the RNA actually can change. So for example, if you give a drug to a person or to a animal subject, you can actually measure the change in RNA of specific gene, and you can use the RNA-seq technique for this kind of purpose. So you can see the difference between two genes, the expression between two genes using RNA-seq. So for example, this is one of the paper that was produced by our group from Yamagishi Sensei. So in this case, he tried to see the gene expression difference between different conditions in the human infected parasite. So if you can see here, for example, this protein, this gene, this CD163 actually have like a different gene expression if they have in the severe malaria compared to the non-severe malaria. And also a protein in human, which is called CBLB, also it's like expressed lower in older age compared to the younger age. And in malaria, in parasite malaria, you can in the malaria parasite you can also see the difference in the gene expression. For example, this pyruvate kinase actually has higher expression in a younger age compared to the older age, and also this early transcribed membrane protein also expressed lower in the older age compared to the younger age. And this result actually contributes to how the malaria can actually be severe in younger age. So you can see those kind of, you can see this kind of experiment can be performed using transcriptomics analysis. And this is also one of the results using transcriptomics analysis, which from my recent paper. So in this case, I wanted to see the gene expression in the dengue infected, the difference of the gene expression in dengue infected compared to the non-infected mosquito. So I found that there is a family of gene that was that actually have mixed expression in dengue infected mosquito. However, these 9 genes in the family has a higher expression in the dengue infected mosquito. So how can we follow up with those research? So we can use this kind of experiment to see what L2, so the gene is called L2EFL. So we can see how the L2EFL actually works in the mosquito by using confirmation analysis. In this case, I tried to infect a mosquito cell using the dengue virus, and I can see that I could see there are actually upregulation of the L2AFL gene expression, which is actually in concordance with my RNA-seq experiment. So I can try also to silence the L2FL. So by silencing the L2FL and infected the mosquito cell using the dengue virus, I can see that the dengue virus dengue virus replication is actually enhanced following the silencing of the L2FL gene. And on the contrary, if I try to upregulate the L2FL expression and then after that I try to infect the cell using the dengue virus, you can see that the dengue virus replication is actually suppressed after try to upregulate the L2FL expression. So then we can actually try to understand that probably L2FL is one of the antivirals in Aedes aegypti. So you can do this kind of experiment if you do the transcriptomics analysis. Without doing the transcriptomic analysis, it would be difficult to do this kind of experiment. So that was one of the reasons that you do transcriptomic analysis. And transcriptomic analysis has a advanced, it has been advanced to a single cell transcriptomics analysis. So this one is focused on gene expression on individual pathogen of cell in a different condition. So if the previous transcriptomic analysis only focus on a different condition, but this one actually focus on a different cell or a different pathogen. And there are this figure here, it shows how you can actually do the single cell transcriptomic analysis. So one of the company that is one of the platform that is major player in this field is called the Chromium platform from the 10x company. So maybe you can look at it later if you want to know how it works. But how you do that? How do you analyze that single cell transcriptomics result? For example, in this figure, you can see here that there are actually a different color and also a lot of dots. So each of these dots, it means a cumulative gene expression of one parasite. So you can see here there are a lot of parasites that has been sequenced individually, and each stage actually give like a different gene expression. So you can see here like this pink, these are all the gene expression in the merozoite form. For example, if this is a cumulative gene expression of a male gametocyte, which is actually different with the female gametocyte. So you can do this kind of analysis to see, and in this figure, this is also another figure of every life of every stage of a malaria parasite. So using this kind of analysis, if you try to sequence one pathogen of one parasite, and if you try to map the cumulative gene expression in this one, you can actually see that which stage the pathogen is on the blood. So this is they do the experiment by sequencing the synchronized parasite for each hour of post infection of the inside the erythrocyte, and you can see that every hour they actually give a very different gene expression, which is actually in concordance with how it evolves inside the parasite. So you can do this kind of experiment by using the single cell transcriptomics. And also this is also one of the application of single cell transcriptomics, which is very common to do, is to see the T cell receptor and B cell receptor. For example, in this one, have a lot of dots in this one. This is one dot equals to one T cell. And you can see here that that CD4 and CD8 actually have a very different gene expression. Not only that CD4 and CD8 are very different, but the subset of each CD4 and CD8 is also very different. So using this kind of experiments, you can understand that the bulk sequencing, bulk RNA-seq sometimes is not enough to do the further analysis. And then we try to move on the epigenetics. So epigenetics is also another hot topic in next generation sequencing. So this research field studies the regulatory system that are heritable, which is not related to the genome. So two cells that are actually genotypically identical, such as the brain cell or the lung cell or the liver cell, they all have like the same DNA. They also have the same genome. However, sometimes, but the gene expression of the brain cell and also lung cell and also liver cell are very different. It's because their epigenetics modification are different between those cells. So this is you can see in this figure here. This is DNA in this one, and this one in chromosome. So in our chromosome, the DNA are not linear, so they're actually built in what is called as chromatin. So chromatin is a collection of nucleosome, and one unit nucleosome contains 8 histone protein and DNA. So in our chromosome, the DNA is actually packed so dense using in the chromatin system like this one. And if you modify the histone protein in this one, by using methylation or deacetylation or acetylation or demethylation, you can actually control the gene expression. So that is one of the factors how our brain cell and our liver cell are very different in gene expression, is because one of the things is because their epigenetic arrangement are very different. For example, in this one, if you see in this graph, you can see that if there are methylation in lysine protein lysine 9 of a histone 3, then the chromatin will be like very dense. The transcription factor will be very difficult to access the transcription start site, and gene expression will be silenced. On the other hand, if there are acetylation in the lysine 9 on the histone 3, the chromatin arrangement will actually be less dense, and in this case, the transcription factor can actually access the transcription start site and start the gene expression. So how is it translated in the real life? For example, this is one or this research found out the epigenetic marker change in intraerythrocyte in Plasmodium falciparum. So they see that in chromosome 2, around this position, on 16 hours post-infection, there are enrichment in this histone modification pattern. So there are actually a lot of histone modifications. So there are about 13 histone modification that they look. Then they see that there in 16 hours for infection, the histone mark of this position is actually enriched. And if you try to find out, if you try to follow about 8 hours, 16 hours, 24 hours, and 32 hours, you can see that the acetylation in the lysine 16 of histone 4, they actually have like a different enrichment type, which actually controls the gene expression on each time point. So this is one of example of epigenetics research. Okay, and then we start with microbiomics, which is probably the purpose of this webinar. So in my opinion, microbiomics is actually a targeted metagenomics. How is it called targeted? It's because we are trying to see the bacterial composition by amplifying and sequencing a universal marker. In this case, for bacteria, there is a universal marker which is called the 16S ribosomal DNA. And if you amplify this one and follow by sequencing, you can finally see the difference in bacterial composition. And in case of fungi, you want to amplify the ITS gene because this is known as the universal marker for fungi identification. However, for using a targeted metagenomics, it's still very difficult for virus because virus doesn't have any universal marker. So this is one of the project that we did in National Institute of Infectious Disease is to compare the microbiomics of stool of HIV infected and non-infected from the Ghana people. So in this case, you can see that in the HIV negative, and this one is HIV positive, the microbiome composition, the bacterial composition in stool is very different. In the HIV negative, you can see there are a lot of Faecalibacterium. However, in the HIV positive, you can see that there are actually a lot of Stenotrophomonas, little to non-Faecalibacterium, and also upregulation of Achromobacter in HIV positive. You can also see that in like some kind of bird's eye view, you can see that the microbial composition between these two conditions are very different, and the diversity of HIV positive actually lower compared to the HIV negative. So this kind of experiment can be done using the microbiomics by amplifying the 16S ribosomal DNA and followed by sequencing. And then there is a metagenomics, which is actually very recent application of next generation sequencing. In this one, you don't need to amplify the universal marker. So you just run you just get DNA and you just sequence all of them. And by bioinformatics analysis, you can see the composition of bacteria, fungi, and also you can do this with virus because you will sequence anything that is in the sample. However, one of the problem is metagenomics is because sometimes the concentration of the pathogen in sample can be very low and you don't amplify the sample. So that's why sometimes you cannot analyze it very clear using metagenomics analysis. And also the host DNA such as human DNA or cattle DNA in your sample can also complicate the result. So that's why metagenomics is still very recent. There are still not many tools that can actually analyze metagenomics analysis with very clear, and especially the DNA have to be depleted from the host DNA and other stuff. So at this moment, I told earlier that there were actually that were major players in the in the next generation sequencing. However, now only Illumina that actually dominates the high throughput sequencing technology now. So there are a lot of systems in Illumina, NextSeq, HiSeq, NovaSeq. These are all very expensive. For example, NovaSeq can output data up to 6 terabase. So using NovaSeq, you can sequence 20 human genome in one sequence. The higher, the more expensive the machine, the lower the nucleotide per price will come out. So the cost relatively will be cheaper if you try to sequence using NovaSeq. And Illumina also not only goes bigger, they actually go smaller in this case. For example, this i6-100, this is the smallest Illumina platform that you can purchase now. It has the output of data of 1 to 1.2 gigabase. This is not enough for human sequencing, but however, if you want to sequence malaria parasite, for example, you can sequence up to 200 malaria parasite using this i6. So it depends on how big your DNA is. This is the choice of the machine that you can choose for Illumina. One of the characteristics of Illumina, they are very accurate. They are very accurate, but however, they can only sequence a short fragment. For example, my 6 series here, they can only sequence up to 300 base pair. So and then we came to the long read sequencer. What makes this long read sequencer are different with the previous Illumina is because they can actually sequence as its name, their names say there are a long read sequencer, so they can sequence very long fragment of DNA without any fragmentation. So for example, the Oxford nanopore Minion can sequence up to 1 megabase on one DNA strand. However, the downside of this long read sequencer, there are have like very high error rate. For example, Minion have about 5% to 15%. However, if we try to pile up all those raw data and try to get the consensus sequence, Oxford Nanopore said that the lower rate can be actually reduced to up to less than 1%. So this 5 to 15% error rate is the error rate of each DNA strand. So if you pile those up and you can get the average, the lower rate can actually be reduced. And the price, for example, for nanopore is very cheap. It's relatively cheap. For $1,000 US, you can start your sequence compared to the previous Illumina, which is very expensive. And Minion also have another advantage. They are very small, so you can bring it like in your field and you can do your sequencing there. They have additional accessories. For example, this is called Minute. This is a CPU, so you don't have to use your laptop to do your sequencing. And they also have the MK2C, if I'm not mistaken. So this is actually have a display by itself. So by using this one and you bring the field, you can use your sequencing like in the middle of a paddy field or maybe in the cave, and you can just do your sequence like on the spot if you really need to find out about the result.
I'm sorry, Dr. Lucky, 5 minutes more.
Yes. Oh, so fast.
Yeah.
So then let me start talking about Minion then. So this is one of the our project with Minion. For example, the systematic anti-malaria resistance detection. So you can use Minion sequencing to find out about the drug resistance by checking the SNP of genes that are related to drug resistance. For example, these 9 genes are related to drug resistance. And if we try to look at the SNP of this gene, we can invert the drug resistance. For example, this sample have all 10 sample have like resistant to chloroquine and suldox, but we didn't know about artemisinin because we use this old chemistry of a Minion sequencer. However, the new chemistry of Minion sequencer are very good. For example, in the sample from Thailand and Vietnam, we can see the K13 mutation, which is related to artemisinin, which are different between Thailand and Vietnam. And also we can see the CRT, which is related to chloroquine resistant. And this also another sample from Manado. We see that there are a very unique mutation in K13, but it is not related to artemisinin because this is not in the non-propeller region. And also we can also use Minion, for example, to genotype. In this case, we try to serotype the dengue virus using Minion. In this case, we collected samples. We amplify the specific dengue virus serotype. There are very we amplify the genome section that are characteristic of serotype using isothermal amplification, and we use Minion to sequence. And you can see here that we in Manado there are a majority of dengue 1, and in Hanoi, Vietnam, the majority are dengue 3, but there are huge proportion of this that we cannot see the serotype, which is actually related to the viral titer. So the lower the viral titer, the more difficult Minion to sequence, to know the serotype. And also this is a targeted metagenomic sequencing with Minion that we did last year with One Health, Gadjah Mada University. So last year we went to Yogyakarta to give training and to do real sequence using sample from the cattle blood. And one of the sample actually gave a good result. For example, this sample actually find out that one of the sample that we sequence have Theileria orientalis as the major pathogen in those samples. And also we can also use Minion to sequence whole genome. So this is whole genome sequencing is beneficial. In this case, you don't have to use PCR to amplify a gene of interest. You can just sequence a whole pathogen, and you can do that with, for example, with Neisseria meningitidis to see the serotyping of this Neisseria. So Neisseria actually have a different marker gene to see whether there are belongs to serogroup A, B, C, W, Y, and C. And if we try to map the sample to this gene, which belongs to the serogroup A marker, it was not mapped, so it was on serogroup A. And this one also, if we try to map to the serogroup B, it was not possible because it was not serogroup B. And this one also was not serogroup C. And it was also not serogroup X. But however, there were confusion whether they belong to serogroup W or serogroup Y. In this case, we have to see that serogroup W and serogroup Y actually, even though they have like a similar gene, there are different places that are actually have a different nucleotides which belongs to each serotype. And by calling the consensus sequence of the previous sequence data and try to map to the serogroup Y gene, we can see that it was a perfect match. And if we try to map to the serogroup Y reference genome, we can see there are a lot of mutation in this one. So we can actually conclude that the serotype was belong to serogroup W. And also another project that we did last time with the with INARESPON is to see the drug resistance of a Microbacterium tuberculosis. So this one in this table, you can see here, these are the genes that are related to each drug. And you can see here that if you want to amplify each gene, using PCR, it would be very time consuming. So that's why you do a whole genome of a Mycobacterium tuberculosis and you analyze it later. And you can see, for example, if you try to map in the RPOB gene, which is responsible for the rifampicin resistant, you can see there is mutation here. And if you try to map also to Gadjah, there is mutation here. But there are no mutation if you try to map the isoniazid A, which is ENHA, which is responsible for isoniazid. And as a conclusion, you can actually comprehensively try to detect the drug resistance, just in this case, SNPs, which is probably are related to a drug resistance by only sequencing once and without any PCR. So you can see that it's actually how sequencing can actually be very beneficial in this kind of application. Okay, so as a conclusion, sequencing method has evolved significantly, and now sequencing method is able to assist like sophisticated research, not only genomics, but beyond genomics. And now you can sequence is, the cost has been like reduced significantly. But to start the sequencing by itself, because we have to purchase the investment, the machine and the region, it can be very expensive. And for that purpose, nanopore sequencer is actually very attractive because not only it's small, it's actually relatively less costly. So it's very perfect to do the sequencing in developing country, and also if you want to sequencing right in the field, such as in the cave, for example, if in this case a bat, to sequence the bat metagenomics. So nanopore sequencer also not only can be applied to genomics, but you can actually use it for transcriptomic analysis and also epigenetics for, and also, so it's still evolving, but there will be another application for nanopore sequencer also beyond genomics. So sometimes people said that nanopore sequencing, long read sequencing, in this case, nanopore and PacBio is actually the third generation sequencing. Okay, so with that, I would like to acknowledge my my colleagues from University of Tokyo, from Hokkaido University, from Münster University, from Manado, Samratulangi University, from Yogyakarta, and from Vietnam, and Mahidol University, and also Inarispon, and where I'm working now at the NIIB. Okay, thank you so much for the the opportunity to present.
Thank you very much, Dr. Luki, for the very awesome, I have to say very and awesome presentation that we learned a lot. I'm sorry for making you run fast on the last 5 minutes. Actually, I don't want to stop you, but we don't have a lot of time here.
Okay, that's okay.
Maybe we can arrange for another webinar for everyone so we can learn more from you.
Okay,
so
sure.
Yeah, I would like to try to conclude all of what Dr. Luki just teach us or share his knowledge that we know since the discovery of the sequence technology. It's decreased a lot of price, and we can get a lot of information as the comparisons. It needs 10 years, more than 10 years for us to sequence the human genome, but since the discovery of the NGS, it can fasten the speed, I think, that we know. At 2014, more than 200,000 human genome already sequenced. That's very awesome. That's a very good technology, and we know that by using the NGS and other sequencing technology, we can do a lot of projects such as the genomics characterizations, and in the transcriptomic, the epigenetics, and the microbiomes, metagenomics, and many more. And we also can do some analysis for the drugs, multi-drugs resistance pathogens using the SNPs and combine it with the NGS sequencing. Thank you very much. And for all of the questions, we will answer it and discuss at the Q&A session. And I would like to remind for all of the participants, for those who have a questions, please ask your questions through the Pigeonhole link that we share in the chat room, so that everyone can see all of your questions. So please not write it in the Q&A column. Thank you very much. So the second speaker, hello, Johan. Are you still
Hello.
there? Thank you
Yes,
very
I'm
much.
still here.
Thank you very much to wake up very early there and come to our webinar.
No problem, no problem. It was not too early. It was 7 in the morning, so not like previous time where it was much more earlier. Anyway, it's good.
Glad to know that. Okay,
Yeah.
so before I give this screen to you, I would like to introduce you to our participants. So, Dr. Johan R. Michaux. He is the research director to the Belgian National Fund for the Scientific Research and the research associate to the French CIRAD, or International Center for the Research in Agronomy and Development. He is also the founder and the head of the Conservation Genetics Laboratory of the University of Leige, Belgium, which is notably expert of the conservation of critically endangered species such as African lion, polar bear, European wolf, European otter, snow leopard, Pyrenean desman, and many more. And he is also working on the relationship among wild mammal, domestic animals, and humans in a One Health and an eco-health approach. These research are based on the different molecular biology technologies like a metabarcoding approach and next-generation sequencing technology in order to screen a large panel of pathogens such as bacteria, virus, or macroparasites. These analyses help to better understand the dynamics of pathogens and spillover risks among the different wild and domestic species, as well as humans. These research are notably developed in the close collaboration with Prof. Wayan Artama and Dr. Wisnu Norcahyo from Universitas Gadjah Mada, and Dr. Johan Michaux already published more than 150 international peer-reviewed scientific articles and collaborate with more than 60 colleges working in 4 continents, such as Asia, Europe, Canada, and Africa. He is also an academic editor for 3 international scientific journals, and one of them is the PLOS One. So, okay, I will give the screen to you, Dr. Johan. Your time is 35 minutes from now. Thank you very much.
Thank you very much. So I will try to add my screen. Okay. Okay, can you see my screen?
Yeah, will coming.
Can you see my screen?
Yeah.
No.
It's okay.
You can see it clearly.
It's okay. Okay. So perfect. So first of all, I would like to thank Professor Wayan and Putu for this kind invitation. It's always a pleasure to talk with all of you and to share many different studies. So again, many thanks. And so I hope that this talk will interest you. And so I would like also to thank Dr. Lucky, because my talk is quite complementary to his talk. So we did not talk before together, but it's perfect because I think that the talk are very, very complementary. So it's good. So yes, so today I will focus my talk on this next generation sequencing methods, and particularly to study the microbiomes using metabarcoding approaches. And so I will start my talk just to summarize a journey of field biologists working also in the fields of genetics. So it will be a first step to explain you how we can use this next.
I think something problem with Dr. Johan. Okay, he's coming again. Hello, Johan. Can you hear us?
It's connection
Hello?
problem.
So for while we waiting for Dr. Johan fixing his connection, I would like to remind again for all the participants, for those that have a questions, please ask your question and write down all of your questions to the Pigeonhole website through just click the link in the chat room, and then you can write down. First, choose the which one the presenter that you would like to ask, or put the name as the tags of your questions, so that we know that to whom you ask your questions. Okay, maybe there is something problem with Dr. Johan. Maybe we will wait. Or maybe if we don't, if it takes a little bit longer, maybe we can start answer some questions that already appear in the, maybe we can start in the Q&A chat box, since it's already have 5 questions. So I will give the screen to Ezel. You ready, Ezel?
Right.
Maybe we will wait for the Dr. Johan.
Okay.
Thank you.
We have 5 questions currently pending for more, and it's towards Mr. Luki. And the first one is from an anonymous attendee, which says, okay, but has her name. Hello, I am Nadia Shukat from UNER. I have to ask Sir Luki, while using PCR, why is it necessary to produce cDNA first? Why not directly measure RNA expressions? Thanks. Maybe Mr. Luki, you may answer.
Okay, so regarding this question, if we want to use Illumina as our platform for sequencing, they haven't actually produced regions to do the RNA sequencing, and it's also impossible to do cDNA sequencing with the to do the RNA sequencing using Illumina without converting it first because to cDNA, because the chemistry is not compatible. And also the principle of the machine itself, Illumina, is actually using a bridge PCR for doing that. So it's just impossible for Illumina. However, if you try to use the sequencing with nanopore sequencing, nanopore sequencing is compatible to do the sequencing using direct RNA. So in this case, if you use the RNA kit to do the sequencing with nanopore, you do not need to convert it first to cDNA. So you can directly use your RNA for using the sequencing. So it depends on your platform for sequencing. Illumina is not possible to do RNA sequencing directly. However, nanopore sequencing, you can do it with the RNA directly.
Okay, thank you for the clear answer. So it's more towards the platform that's being used. As we can see, Mr. Dr. Johan is back, so the floor is back to you, sir.
Thank you. So do you hear me? I don't know what happened. So I will start again, and I was seeing you, but you did not see me.
Yeah, Johan, you can share.
Okay, can you see my screen? Yeah.
Not yet.
Can you see it? Can you share my? Can you see my screen?
Okay. Yeah. It's processing.
Okay. I hope that I will be continue. So I was saying that it's okay.
For me, it's still processing. Maybe we can wait just to.
Yeah. Okay.
Yeah, it's appeared. Okay, it's perfectly.
Okay. Okay. So I will start my talk by explaining how the next generation sequencing is working and how it is possible to perform metabarcoding methods. So First, this first part of the talk will be divided in different steps. So first, I will just give a few information about the field work, because it's very important if you want to work on wild animals. After, I will explain how to extract the DNA, how to perform the first step before the sequencing, for the sequencing itself, and after how we can identify and analyze the raw sequencing coming from the automatic sequencer. And so after, I will also show you different kind of data visualization. So yes, the first step, can you see my screen? It's okay?
Yeah. You can see it.
Okay. So yes, the first step, of course, when you are working on wild animals, so it can be here rodents, but it can be bats, it can be elephant, it can be many different species. So of course, the first step will be to identify which kind of bacteria or viruses you want to study. And here, for example, on rodents, you can kill these animals because they are not protected, and you can choose different kind of tissues, like the liver, like the spleen, like the kidneys, like the brain, etc., according to the bacteria communities or virus communities that you want to study. So after that, when you come back to from the field with your samples, you will have also to
He's gone again. Okay.
okay.
Hello?
Yes, can you hear me?
Yeah. But I cannot see
I'm
you.
sorry. I have my screen is still shared. You don't.
No, we cannot see your share screen. Okay, this is processing now.
I'm sorry, I'm at home because of the lockdown. My Wi-Fi is not good here, so maybe it's the reason why I have a problem. So
that's okay. Maybe you can turn off your video to make the bandwidth of your internet becoming stable.
Yes, maybe I will switch on.
Yeah,
Switch off.
sometimes it works.
Yeah.
Yeah, now we can see your screen.
You can see my screen. Okay. Zoom does not want to see my face.
It's just gone.
It's okay.
Yeah, it's processing now.
Okay. So can you see?
No, but it's still processing. Yeah. Okay. Perfect.
So anyway, if you want to study microbiomes, and if you want to work with next generation sequencing, one of the main problem is the contamination, because as this method is very sensitive, you can have very quickly contaminations among different samples, for example. So you will have absolutely, if it's possible, to work in a sterile environment to absolutely remove the external contamination, to homogenize the sample in order to have the maximum of bacterial species were in sample. And of course, we'll have to add negative controls. It's very important in order to check the quality of your analysis. And if it's possible, it's all testing user plots, because you do not manipulate a lot your sample, so you avoid to have any contaminations. So it's okay.
Oh, I'm sorry, everyone. We didn't expect this one. Hello, Johan.
Terrible. Hey, yes. Can you hear me?
Yeah, I can hear you.
Okay, maybe I will not use the presentation. Maybe can you see my screen?
Yeah.
It's still processing. Okay, we see it.
I'm very sorry. Okay. So after extraction DNA step, you can start the pre-sequencing step, which in fact correspond to two PCR. And between the two PCR, you have different purification steps and also decontamination. So when you're working next generation sequencing methods, in fact, the first PCR will be to amplify the fragment that you are using. So for example, for the study of bacteria, you work with a small fragment of the 16S ribosomal gene, and this 6S ribosomal. Okay, can you hear me?
Yeah.
Yeah.
Do you need a little bit time for you to fix your
Maybe,
connection?
yeah. Yes, maybe. Yes, maybe I will switch off and switch on my Wi-Fi box, and so during this time, maybe you can answer to different questions.
Okay.
I hope that it will work a little bit better. I'm sorry for the problem. I'm just come back in 3 minutes.
Okay. Thank you very much, Johan.
Okay. Okay. All right.
Yeah. So everyone, while we wait for Dr. Johan fixing his connection, I will give again the screen to Ezel, so we can start answer some questions with Dr. Luki. The screen is yours, Ezel.
Okay. Wait, let me put this on full screen. All right. And then you guys can see my screen, right?
Yeah.
Yeah, perfectly.
All right. Perfect. Is Mr. Luki. Okay. Okay, this is from an anonymous person, and the question is, I used the extraction kit which is special for microbiome analysis. This kit can deplete the host DNA. However, we failed to get the high concentration of DNA. Can you give some advice?
I'm not sure actually. There are a lot of factors that can affect if you lose the high concentration of DNA. So I'm not sure what kind of extraction kit do you have. So I can't really give some kind of advice, but some kit that can actually deplete host DNA is probably not. It's probably not compatible with gram-positive bacteria. So for example, there is one paper that I try to follow how to deplete host DNA using saponin. In that case, the host DNA was depleted, but gram-positive was also depleted at the same time. So you can actually get a low concentration of DNA. So in this case, I'm not sure the extraction kits, so I cannot give you some advice. But you may try to like look for the troubleshooting maybe in the manufacturer protocol or something.
All right. Okay, we'll go to the next one. If the question has any more clarification, maybe you can chat into the chat box in yeah. Okay, we'll go to the next question, and it's towards Doc Mr. Luki again, and it's from Aji Purbayu. Is it possible by the latest sequencing technology to estimate area of mutation and predict the mutation effect to the genotype of the pathogen or virus?
Okay, so I'm not sure what does AG mean with area of mutation, but the latest sequence technology can basically see all the mutation that happens in one sample. They have like enough power to do that, to do the sequencing for all the whole genome, and then you can try to see the mutation. However, if you want to predict the mutation effect, how the phenotype of a certain genotype, it will not be possible to just look at the genotype. You have to do some validation analysis. So for example, if you find a mutation that interests you, you can try to reverse it back to like the wild type using some kind of molecular biology technique. And you can see the effect after you on the cell or the animal study that you do after you reverse the mutation, the wild type. So it's not possible to see to predict the mutation only using looking at the genotype.
Okay, so it's more like towards focusing on certain parts of the genotype. Is that correct? Or
Sorry.
it's okay. We'll go to the next question.
Okay.
So this is from Aji again, and it's towards Dr. Runtuwe. If I found a protein and I wanted to know which gene that encode or control the protein, how I should do generally.
So for one, if you found a protein of interest and you want to know which gene that controls it, you have to first try to find out the sequencing of the protein by using protein sequencing. I'm not very familiar with protein sequencing, but I think you can do like some kind of mass spectrophometry or de novo protein sequencing, and you can see the sequence of the protein. After you found the sequence of the protein, you can try to map it to a database, and you will such as you can blast it using a blast with the protein, and you can see which gene that actually controls the protein of your interest. So first you have to find out the sequence of your protein by using the protein sequencing. And after that, it would be probably very easy to see which gene that actually controls the protein.
Okay, thank you for the answer, and we'll go to the last one, I believe. So this is from Sina from UGM, and the question is to Mr. Rutune. I just wanted to know, is it possible to use Minion to sequence DNA from the environmental DNA, eDNA samples, just for example, to sequence DNA in the feces of an animal?
Oh yeah, it's possible to use Minion for this kind of purpose. In fact, we just actually did some kind of experiment. I did not present today, but we tried to do the environmental sequencing from toilet water and also from the kitchen sink to see the bacterial composition that the bacterial composition in those kind of environment, and we can actually see the using the metagenomic sequencing using Minion. So we can actually see the bacterial composition in such condition. So it's possible to use Minion for such purpose.
All right, thank you for the great answers, Dr. Luki. And I believe Mr. Johan is back online.
Yes, I'm back again. So I deeply apologize for this problem. So now I will work with my 4G connection with my mobile.
Thank you very much.
I hope that the situation will be better.
Okay, thank you.
No, I'm sorry, it's me.
No, that's okay. That's okay. It's always happened for internet connections, especially in these conditions.
Yes, it's a very big problem with the lockdown. Can you see my screen now?
Perfectly.
Very clear.
Fantastic. Many thanks, the 4G. Many thanks. Okay, so I am coming back to my talk. So I was explaining, that when we are working on metagenomics, so there is different steps. So first steps, of course, is in the fields. So you have to select the tissue that you want to study according to the pathogens that you want to analyze, bacteria, viruses, etc. After that, you are coming back in the lab. And so you will have to be very cautious when you will extract the DNA, because the content contamination can be a big problem when you are working with such kind of technologies. And after, when the DNA will be extracted using, for example, one robot or at least one rare DNA room, you will be able to start the PCR. And in fact, with the Illumina sequencing method, we are using 2 PCRs. So the first PCR will be a quite normal PCR, so it will allow to amplify the fragment of gene that you want to analyze. So for example, here for the bacteria, we are using a fragment of the 16th S ribosomal gene, which is very useful to study many bacteria communities because we have a very good database in the public databases. And this first PCR will be a little bit special because we will associate to the amplified fragment a small adapter that you can see here in blue. And in fact, this adapter will be very important because it will allow you in a second PCR to associate different tags. And these tags, you know, that will be associated at the forward side, but also at the reverse side. These tags, in fact, will help to identify the sample that you will study in your analysis. And the interest when you are working with Illumina technologies, for example, is that you can analyze, for example, more than 500 different samples at the same time in the same run. So it's very interesting, of course, to identify each of these samples. And so it is quite easy because with a few number of tags, in fact, you will be able to analyze an important number of samples. And so here, for example, I give you, you know, a plate of 96 samples that we are generally used in the molecular biology laboratory. And in fact, you know, for example, we will use 12 different tags. And so, for example, when we will associate these 12 different tags on our 96 samples, for example, this first tag will be associated in the forward side of the fragment, the amplified fragment. But we can also associate different tags which will be linked to the reverse part of the fragment. And so according to these different combinations of different tags, you know, you will only need of 20 different tags to have 96 different combinations, which will allow you to analyze, for example, here, 96 different samples in the same run, in the same analysis. Okay, so it is just to explain you how such kind of metagenomic, meta barcoding method are working. So between these different PCR, of course, we will have to perform different purification steps. This will allow you to discard any primers and primer dimers, for example. And so there is different kits to purify our PCR products. And for example, on our side, we are using magnetic systems and so magnetic beads. And so in fact, such kind of methods is working perfectly. So before to perform the sequencing, we will also have to perform DNA quantifications because in fact, all the samples that you will analyze have absolutely to be equimolar. So we have to have the same quantity of PCR product for each of the samples that we will analyze. So this is the last steps of the last step before the sequencing. And then after, we will be able to perform the next generation sequencing analysis. So there is different techniques, the 454 pyro sequencing, which is now quite old and which is not used, the ion torrent, and as Dr. Lucky explained, we have now many different kind of Illumina sequencer according to the number of sequence that you want to obtain in your final results. So you can have, for example, the high-sec sequencer, which allow you to obtain until 3 billions of sequences of different sequences. And now we have, for example, the NovaSeq and the NextSeq methods, which allow you to have at least 5 to 600 millions of reads. So you will have, after the sequencing, many different sequences. And so it will be very important after to clean all these sequences, because many of them could not correspond exactly to what you want, but it could correspond to different artifacts. So in fact, you will receive, a file, a FASTQ file, which will give you millions of sequences. And so for each of these sequences, you will have 3 different information. First of all, the sequence ID, which give you the identification of the sequence. You will have the sequence, of course, and you will have also different other information about the quality scores. So it corresponds to the run, the sequencing run. And so after that, we will have to clean all these sequences in order to obtain and to analyze only the best sequences and to remove all the artifactual sequences. And so for that, we will have to use all the many different pipelines, bioinformatic pipeline. So just a summary here of this script, bioinformatics script. But just quickly, this script, in fact, will allow you to assemble the pair-ended sequences, so the forward with the reverse sequences of the same sample. It will allow you to discard too short or too long sequences, which will correspond to different artifacts. This will allow you to remove primers on both ends. This will allow you to discard the sequences with low quality scores. And of course, it will allow you to discard any chimeric sequences. And so after that, after all this cleaning of these millions of sequences, you will be able to attribute the sequences to their corresponding taxon. So in fact, you will perform a kind of blast. So you will compare all your sequences with the public databases or private databases. And so this will allow you to identify all the sequences that you obtain. And so for example, here we worked on the Lyme disease, but so I will come back later. And so at the end, you will have a kind of table. And so for each sample that you analyzed, you will have different kind of OTUs. So OTUs is operational taxonomic unit. So it corresponds to a species of bacteria or of prey if you are working on the diet of animals, etc. And so at the end, you will have a list of different bacteria species, for example, for each sample that you study. And so after you can have different kind of data visualization. So for example, the most simple is a pie chart. So, you can evidence here the different families of bacteria that you obtain in your sample. You can also use a heat map. So heat maps are very interesting because, each column corresponds to one sample. And so each line corresponds to one kind of bacteria. And so according to the color of the heat map, you will be able to see if the bacteria family or genus is present in your samples. For example, here in black, you have absence of one bacteria, and in more you are going to the red, more you will have an important presence of the bacteria. And so according to the different sample, you will be able, for example, to identify different kind of bacteria communities. So is it clear for you? Is it okay?
Okay.
Okay, so now I will start to give you some examples. So we have numerous examples. So I was thinking at first to talk about the bats studies that we are performing, but in fact, I will give you also different examples because we have more precise results for the moment. So one of the first example corresponds to the case of the pathogens which are transported by the ticks and the birds in Europe. indeed, in Europe, in fact, every year we receive many birds coming from different regions like the north of Europe or Africa. And so they are coming each spring in Europe after their migration. And so sometimes they are not coming alone, but they can also come with different parasites. And so one of the main parasites is ticks. And so you can see, for example, these different bird species with different ticks.
Yeah.
And of course, the ticks are a very big problem because they can be vectors of many pathogens, like the Borrelia bacteria, which give the Lyme disease, which is a big problem in Europe and in the States, for example, the Q fever, tularemia, tick-borne encephalitis, etc. And so the questions that we had on this study was which tick species are transported by birds, because there is numerous species of ticks. What is their geographic origin and which kind of bacteria are transported on these ticks? And so we performed next generation sequencing analysis. So I will not come back to all these steps that we performed. And these analyses were performed using two kinds of markers. So the COR1, the cytochrome B, the cytochrome C oxidase 1, which allowed us to identify the tick species, and the 16S ribosomal region to perform the bacteria barcoding. So again, we purified the samples, we quantified, etc., and we performed the next generation sequencing analysis. So the results allowed us, on the tick point of view, to identify at least 3 different species, which are very different. And we also identified, for example, 2 different lineages of one species, which is called Ixodes ricinus, and which is the main vector for the Lyme disease, for example. The next generation sequencing method is very useful as compared to the Sanger sequencing, for example, because you can mix different samples in the same sample. And you can also analyze a very small larvae, for example, which have a very small quantities of DNA, but with the power of this sequencing method, you can obtain sequences. And so for the bacteria, for example, we evidenced at least 90 different genera of bacteria. And so we evidenced different genera of interest, like Borrelia, Rickettsia, Ehrlichia, etc. And so we evidenced, for example, different symbiotic, many symbiotic bacteria, but also different pathogenous strains. And so it also, such study it also evidenced, for example, the biological influences on the bacteria's distribution. And so I will not enter into detail, but when we compare, for example, the influence of fed or non-fed ticks, or the influence of the season as considering the different, for example, bacteria genera, we can see, for example, that according to the season, you know, here it's in red you have autumn, and in blue you have the spring. But in fact, you have different kind of bacteria families or bacteria genus. For example, in autumn, you will observe more Rickettsia, Rickettsia bacteroides, etc. And in spring, you will observe a much more diversified quantity of genera of bacteria. So, for example, here in this table, we can also see the link between the tick species, the bacteria prevalence, and different environmental traits. And so here, for example, if we consider only the Rickettsia bacteria, we can see that the prevalence is different according to the species of ticks. So, for example, here we have Ixodes arboricola, Ixodes frontalis, etc. But also the prevalence will change also according if we consider fed ticks or non-fed ticks. So in blue, it's fed ticks, and in red, it's non-fed ticks. We will see also that the prevalence change according to the season. Here we are in autumn, here we are in spring. And so we can see, for example, that the prevalence of Rickettsia, it's much more higher, for example, in autumn for fed ticks. So, you know, you can see how we can analyze such kind of databases. So anyway, I will give you very quickly, as we only have few times now, I will give you another example, which is the story of the Lyme disease in Canada. So we worked on this topic a few years ago, and we work more particularly on the main reservoir of the Lyme disease, which is linked to the Borrelia bacteria. And so the main reservoir of the Borrelia is a small rodent, which is called Peromyscus leucopus. And so the distribution area of this species is quite spread all over the United States, but also in the south of Canada. But with the climate change, we see year after year a spread of this species in the north. And so the question was to analyze, when to simulate what will be the spread of this species in the north of Canada, and according to the spread of the reservoir, also to study the spread of the Lyme disease in Canada. And so you can see here again the small rodents. So the vector to give the Lyme disease is a tick again, which is a typical ticks living in North America. It is Ixodes scapularis, and here the pathogen, you know, the bacteria Borrelia burgdorferi. And in fact, we wanted to analyze two hypotheses which will allow us to test the rapidity of the spread of Peromyscus in Canada. And so there is two main different hypotheses to study such question. So the first one was the diversity disease hypothesis, and the second one was the NMI release hypothesis. So very briefly, the diversity disease hypothesis is based on the idea that more you are far from the center part of the distribution area of one species. Okay, can you see me again?
Yeah.
Okay.
But we cannot see your screen.
Okay. It's okay. You can see my screen?
Yeah.
Okay, so the diversity disease hypothesis is based on the fact that more you are far from the center of the distribution of one species, less the genetic diversity will be important. And so if the level of genetic diversity decreases when you are in the peripheral areas, you can suppose that the pathogen load will increase. Indeed, if you have a lower level of genetic diversity, you can have a lower level of the gene associated to the immune system, and so you will have more risk to be contaminated by many different pathogens, because your immune system will be lower, will be less effective. And so if you have a lower level of genetic diversity and an increasing of a pathogen load, you can expect that the fitness of the animals will be lower also, more you are far from the center of the distribution of one species. And so according to this hypothesis, we would expect that we will have a weak colonization potentiality more you are far from the center of the distribution area. But there is also the totally contrary hypothesis, which is the enemy release hypothesis. And so this hypothesis is based on the fact that as the species extend from its historical distribution range, it will be not necessarily followed by all this pathogen. And so this will result with in individuals which will have a better shape at the edge of the range, with a higher ability to colonize new territories. So just to summarize, following this enemy release hypothesis, more you are far from the center of your distribution area, more your fitness will be better, because you will be associated to a lower number of pathogens. And so if we follow this hypothesis, we will have a high colonization potentiality for the species. So we try to test both hypotheses to better understand the potentiality of spread of Peromyscus leucopus. And so according to the obtained result, we try to better estimate the impact of the reservoir spread to the appearance of the Lyme disease in Canada. So for to obtain such kind of results, we used different kind of tools. So first of all, we analyzed the genetic diversity using different genes which are associated to the immune system, like the major histocompatibility complex genes. We also analyzed different tests to estimate the fitness of the animals. And so, for example, we tested the stress of the animals. We also studied the general fitness, for example, considering the mass size ratio or the sprint size, for example. And again, using next generation sequencing method, we estimated the diversity of bacteria according to the different population that we studied. And so I will not enter into detail, but we analyzed many different populations from the south of the distribution area to the north of the distribution area of the rodent, Peromyscus leucopus. And so we analyzed the diversity of the major histocompatibility gene. And so we evidenced the existence of at least 38 different alleles out of the more than 200 mice that we analyzed from 16 different populations. But our surprise was that we did not see many differences, or at least significant differences concerning the allelic richness when we compared the different populations coming from the south of the distribution of Peromyscus to the north of the distribution area. So it means that we have weak differences according to the latitude if we consider the level of genetic diversity. So we do not observe, for example, the idea that we would have a lower level of genetic diversity more we are far from the center of distribution of the species. So we have evaluated also the stress of all these animals using a serological test of the corticosterone. And again, we did not observe important differences concerning the stress of all these populations. All the animals had the same level of stress, whatever their region of origin, if they come from the north or from the south of the distribution area. We also analyzed the body condition index, considering different aspects like the size, the body, the weight, the spleen, the weight of the spleen, etc. And again, we did not observe significant differences among all the population that we studied. When we worked on the bacteria communities, using again this next generation sequencing method, in fact, you can see here, a heat map that I presented you a few minutes ago. And in fact, we did not observe also important differences of bacteria communities among the central versus peripheral populations. And so the take-home message from this study was that when we compared the central and the peripheral populations, we did not observe differences concerning the allelic richness, so the genetic diversity, concerning the stress of the animals, or concerning the body size conditions. So it means that the fitness was almost the same, whatever the region where they're studied. And we did not observe also important differences between the bacteria communities. And so our conclusions of this study was that there is no reason for the moment to observe a decrease of colonization of Peromyscus leucopus in the north, according to the climate change. And so this have an important link to the spread of the Lyme disease. And since the last 5 years, for example, we observe an increase of the Lyme disease in many regions of Quebec, which is a province of Canada. So it seems that for the moment, the Lyme disease problem will increase year after year in Canada. But we also had another interest of metabarcoding analysis for this study and for the study of bacteria communities. Johan, because we, yes, only 5 minutes?
Yes.
Okay, I have almost finished. In fact, we evidence that this next generation sequencing technologies allowed to detect the Borrelia bacteria, so which give the Lyme disease, in tissues with a sensibility of detection 4 times higher as compared to the classical PCR. And so we propose now for the Canadian administration to study these NGS methods in order to better detect the presence of Borrelia bacteria, and then to give the best medication against this disease for the human populations. So very quickly, another example. For example, we studied the snow leopard in Mongolia, and again, on the basis of faces, for example, we evidenced different groups and genera of bacteria, and again, different pathogenous strains like the Rickettsia, like the Coxiella, but also, for example, the Yersinia, which can give, and particularly the Yersinia pestis. So again, without disturbing this critically endangered animal, just working on faces, we were able to analyze their bacterial communities, and then we were able to better identify their health, for example. And so very quickly, so we are also using these methods to study the Ebola cycle in Central and Western Africa. So this project is developed with European Community Funds. So the name of this project is EboSursi. So we are associated to this project. And the main aim of this project is to better understand which are the reservoir of the Ebola virus, and also which is exactly the Ebola cycle, because for the moment we do not have any idea if the Ebola virus is only living on bat reservoir, or if this virus can also live in different other reservoir, like the small ungulates or the apes, for example. And so the aim of our study is to perform different genetic studies on 6 African frugivorous bats, which were identified as reservoir for the Ebola. So I give you here, for example, 3 different species. And so different studies based on antibodies, for example, had evidence that these fruit bats are the potential reservoir for Ebola virus. And so for the moment, we are studying again the bacteria communities, but also the virus communities using metabarcoding and molecular approach. So this part of the project is developed by our colleagues for the French RID, for example, Martin Peters, which is a colleague. But we are also working on population genetic studies to better understand the mobility of these species, because some of these bat species can have a mobility of more than 1,000 kilometers. And so we suspect that this mobility could spread the Ebola virus from Central Africa to Western Africa. So it's a very big problem in Africa for the moment, because we suspect that Ebola could explode in many different regions of Central and Western Africa. And this was the case 6 years ago, for example, in Western Africa. And so, for example, we are using next generation sequencing method to study the diet also of these different bat in bat species, in order, for example, to better understand if different species can eat the same fruits, for example, and then if such kind of contact through the fruits which favor the spillover of different viruses, like the Ebola virus, from one species to another species. So this project is still in progress, so I hope to give you more details in the future about this project. But so for the moment, we are also working with Professor Johan on another project, which is the case of the rabies and the dogs in Indonesia. So it is a multinational project associating Indonesia and the Gadjah Mada University, but also our colleagues from France and my university from Belgium. And so this study is in progress, and also with Putu, of course, who is also associated to this project. So for the moment, we are waiting that the COVID pandemic stop a little bit to have more accessibility to the fields. But anyway, this project will also associate different aspects, like socio-ecological studies, like the study of rabies virus, but also the study of different bacteria communities, again using the next generation sequencing technology. So we are hoping that this project will be able to continue in the future. And so we also have another project, which is the study of the Sumatran elephant in collaboration with Vishnu Nurkaryo. And so one of the aim of the study is to have a better idea concerning the pathogen relationships among the captive and wild elephant living in Sumatra, but also the relationship with the cattle and the human population living in the surrounding areas. Unfortunately, like for the rabbit project, the research is almost in standby due to the COVID pandemic. And so I'm looking forward that this pandemic finish to be able to continue working on these very, very fascinating topics. And so I thank you very much for your attention. And so I'm very sorry for the Wi-Fi problem. And so I hope you...
It's okay.
Okay. And so if you have any question, I'm here.
Okay, thank you very much, Johan. You're still with us, right?
Yes.
Okay. I'm just afraid that we are lost on you.
No, I'm here. I'm here.
Okay. It seems that your connection, internet connection, is very good now. So,
yes, it's yeah, it's better.
Yeah, thanks God. Okay, so a little bit summations from Dr. Johan's presentation that we learned about step by step about how we conduct the research with the NGS, and then the application of the NGS and metabarcoding itself in the several studies, such as the pathogens transported by ticks and birds in Europe, that we see how it's presented very well, and the Lyme disease in Canada, and the pathogens identification in snow leopard cases. I think maybe while Johan a bit ago, for a while, several time ago, we have a question from Pak Sena, if I'm not mistaken, that he asked about the using of the metagenomic analysis for feces sample. So we have the answer already. And the last is the study of the Ebola cycle in Central and Western Africa, that we can reveal the potential reservoir of the Ebola virus. Thank you very much, Johan, for your outstanding presentations. The internet connection is okay. Everything is good. Thank you very much. So let's move to the Q&A section. We have several questions left. I will give the section to Ezel.
Okay, we'll use the pigeonhole, and then if we have enough time, we'll go back to the Q&A inside the Zoom. Okay. So this is to Mr. Johan by an anonymous person. The question says, should we still do any confirmation tests for identification of bacteria by using NGS, as it could be 4 times more sensitive compared to PCR and may be conventional diagnosis? Mr. Johan?
Yes. And in fact, we propose now to to the medical center to use the NGS method, because it's much more powerful. Because with the Lyme disease, more quick you give the good medication, less you will have problem. Because with the Lyme disease, if you do not treat this Borrelia bacteria quickly, you will have very quickly problem like neuronal problem, like muscular problem, etc. And so the idea is to propose the NGS method in order to have a quicker diagnostic for the Lyme disease, and so to give the best medication as quickly as possible. So we think that it could be a good step which would be followed for the Lyme disease, but also for different other diseases. And so the interest also is that we also evidence that there is a co-infections between different bacteria. And so it's the interest of metabarcoding analysis, because we do not analyze only the Borrelia strains, but we also evidence different other genera of bacteria. And so, for example, I don't remember which kind of, it was, I don't remember if it was Ehrlichia or another problematic strain, which was systematically observed with the Borrelia in people which were contaminated by ticks. And so it's the co-infection between Bartonella also, yes, sorry. It's, you know, the co-infection between Bartonella and Borrelia, which can give many symptoms and which could, yes, be problematic for the health of the people.
Okay, thank you for the very clear answer, and we'll go to the next question. And it's towards Mr. Johan again. And it's, would you please repeat the methods on how to prepare DNA to go to the NGS?
Okay, you mean when you are in the fields or when you are in the extraction room?
Maybe the person who asked can put their answer in the chat column so we can clarify the question.
Yeah. Well, anyway, yes. When you are in the fields, in fact, there is different methods according to the pathogen that you study. So, for example, for the bacteria, it's quite simple because you can only put the tissues in ethanol 96. And all the bacteria will be fixed on the tissue. So it's quite easy, particularly when you are working in tropical regions, you know, where it is quite difficult to have a freezer and where it is quite difficult to have a good logistic. But so it's normal because you are in when you are in the middle of the forest, it's not easy. And for the viruses, for example, we can use a different buffer like the RNA which allow to avoid the destruction of the RNA, for example. And so there is different kind of buffer and of conservation buffer according to the the pathogens that you want to study. But ethanol for the bacteria is perfect. RNA for virus is also perfect. And of course, if you can have access to a liquid nitrogen, for example, it's also very good because you can put directly your samples in the liquid nitrogen. But again, when you are in the middle of forest, it's not easy to have such kind of material. And so when you are in the lab, again, the most important step is to avoid the contamination. So for example, in my lab, we have a room dedicated to the study of rare DNA. So coming from feces or coming from, non-invasive material, for example. And so such kind of rare DNA room is very important. You know, we have a specific material that we only use for next generation sequencing technologies. And even if we are very careful, if we are using robots, sometimes we cannot avoid contamination. So it's a very important step. And so if you want to develop, for example, these methods at the UGM, I would really propose you to create such kind of room dedicated for the rare DNA, because it's a big problem for NGS. I don't know if Dr. Lucky have a similar experience, but it's a big problem if our laboratories.
Maybe if Dr. Lucky has any adding, if he wants to answer extra.
Yes, I actually had a experience with the contamination. For once, where we tried to do the microbiome analysis from the from a blood sample, and actually I found some kind of interesting mycobacterium, which is actually in the blood. So it's actually very rare to find like microbacterium in the blood. But after I found it later, like the bacteria is actually related to the water that we use to extract the DNA. So yeah, with microbiome analysis, it's very contamination is one of the big problem that we have to try to not contaminate our samples.
Mm-hmm.
All right. Thank you for the answers, and we'll go to the next question. And I believe this is towards Mr. Johan again. It's, when we use RNA later to transport an infectious sample of a viral infection, does the virus still have infectivity during the process in the lab?
No, normally not. No. I think that the viruses are fixed. And so I think that there is no risk to be infected. That's not the case if you use a liquid nitrogen, for example. But normally, yes, the virus are stopped. Yeah.
All right. Thank you for the answer, and we'll go to the next question. It's from another anonymous person, and it's to Dr. Johan again. From your experience, how you kept your samples from the field before processing in the lab to reduce the change in microbial position of the sample?
So again, you have to be as quick as possible, and to avoid, yes, to avoid any environmental contamination. So, for example, when we are working on feces, we are trying, for example, to collect the feces directly from the ass. I'm sorry, of the animals, you know, and not when the feces are falling in on the ground, because you can have very quickly contaminations if the feces, for example, are in contact with yes, or the kind of tissues or substrates. So I think that the secret is to be as quickly as possible, and to put the material in a inhalator or in ethanol very quickly. I think it's the secret of the. But of course, we cannot avoid contamination sometimes, and of course, we always have to use negative control on each blank tubes when we are in the fields, when we are doing. Okay, you hear me?
Yeah.
Okay, so you know we always have also a negative test, you know, at each step in the fields, in the extraction step, in the PCR step. And so we can also use mock communities. So it's a kind of collection of different bacteria, for example. And these mock communities can be also used as a positive, how to say, yes, positive control. So we can have negative control and positive control. But again, the problem with the NGS, it is so sensitive that it's again the problem of contamination. But with this negative and positive control, and if you are very careful in the field and very quick, you avoid at the maximum such kind of problems.
Okay, thank you for the answer, and we'll go to the next question. And this is from Miss Mary Ann Devonport. So the question is, could you suggest other database besides BLAST for identifying the OTUS, Mary and Ferry, Universitas Berlin?
It's for me again, or?
I believe it's for you, Mr. Johan.
Okay, other databases beside BLAST for identify those tools. But for the moment, we have this public databases, which is GenBank or Borne or specific databases for the bacteria. So we are using them, but we are also using private databases, which are created in our own institute. And so for the moment, well, yes, there is different kind of BLAST to compare our sequences to the public databases. But again, the idea is to compare the obtained sequences with the public databases. So I don't know what she wants exactly as information.
Okay, thank you for the answer.
Maybe I can add in this.
Oh yeah, sure,
Yeah.
please.
Yeah, in my experience to use to identify OTU, there are actually like green genes or silva database that you can actually use. So this is basically not from BLAST. Is you can have to download it separately. So if you have ever any experience using Chime for analysis, they actually basically using green gene for database. So yeah, there are lot of a database besides BLAST, and what I can say is green genes and silva are two of them.
Good.
All right, thank you for the answers. I believe the time is nearly over, so I'm gonna give back the mic to Mbak Putu.
Yeah, thank you very much, Ezel. And for the last session, for the Q&A session, I think it's already finished. I'm sorry that we cannot continue answering all of the questions, and I believe everyone is very interested with this topic. Even me, also really interested, and I have a lot of questions, but maybe I will meet both of you in the spiriting time. So thank you very much for the Q&A session, and I will give back the session to Krishna Noli. Thank you very much.
Thank you very much.
Okay, thank you, Mr. for becoming our moderator today. And here we are at the end of the webinar today. But before we end the webinar, according to plan, we will give Prof. Wayan will give a closing statement first, and then we can take a picture. Okay, for Prof. Wayan, please give a closing statement for us.
Okay, Noli, thank you for all participants from all over the countries and from abroad, and also for the two speakers, the distinguished speaker, Dr. Luki and Prof. Johan. Thank you so much for you sharing everything to us, but not only today, maybe in the collaboration the next time. I'm waiting for you in Indonesia, and Dr. Luki on GSPS, and also Johan on CDOGC. Yeah. Okay, thank you everybody to join us in the webinar for the last webinar in this year. Maybe next year we will arrange another webinar with a real topic with what we do in Indonesia with both of the participants, our guests from Belgium and from France and from Germany. We have a collaboration also with German scientists and with Japanese. Okay, thank you so much for today, and have a nice day for Johan in Europe and Luki in Japan, and all of us, and also other colleagues from other different countries. Okay, thank you so much for attending this seminar. Thank you. We take picture first.
Yes, take picture first. Okay. Okay, I will take the picture in 1, 2, 3. Okay.
Also, thank you, Esel, for your participation. Esel is our student.
Thank you.
Okay. Okay. Thank you, Prof. Wayan. Thank you for Dr. Luki. Thank you for Dr. Johan for becoming our speaker. Very great presentation today. And thank you for all the attendees today that have attended this webinar. At the end of the webinar, we will share attendee form, and please fill the form to get the e-certificate that will be sent on the next week. Okay. Enough for me. If there is any mistake, we hope. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh. Good night, everyone, and goodbye.
Goodbye.
See you again.
Okay, Johan, thank you so much. Luki.
You are welcome, Wayan.
Yeah.
See you next time, hopefully.
Yeah. After Corona.
Yes. Bye-bye.
Yeah, we just finished collecting sample from Bali.
Good.
Yeah.
I hope to. Yeah.
We got ectoparasite and also blood sample, urine, swab. Yeah.
That's perfect. I'm really looking forward to come back again in Indonesia and to meet all of you. This coronavirus is a big problem. It's terrible.
Yeah.
Anyway, I hope that. Yeah.
Okay, for Luki, our JSPS, please keep contact with us. We will do it. Yeah.
Hopefully we can go to Komodo next year.
Oh yeah, of course, in July. Both of you.
Yes, with pleasure.
Yeah.
Okay.
Bye-bye.
Okay, thank you.
Thank you very much.
Okay, selamat malam, Luki.
Malam, Prof.
Okay, the attendance form will be here for maybe 10 minutes for the attendance in the form online.